Okay, so if you've been following along for my Unit 5 AP Bio playlist, this is our last topic in the unit, so great job. And I feel like it's a repeat of a lot of things you've already learned throughout the unit, so hopefully it should be nice and enjoyable. Anyway, let's go ahead and look at the essential knowledge that is, or there's like 3 basic key points that this topic is trying to get you to understand. So the first one is that segregation, independent assortment of chromosomes, and fertilization result in genetic variation in populations. Now we also have crossing over as well as mutations create variation as well. But here when we talk about Mendels Law of segregation, really what we're talking about is how a diploid organism who has 2 chromosomes, those 2 chromosomes are going to separate or segregate into gametes. And so here, during meiosis, if this male is heterozygous, during meiosis, his 2 chromosomes are going to separate throughout meiosis 1 and then meiosis 2. And so here in his gametes, half his gametes will pass on the allele for brown eyes and half his gametes will pass on the allele for blue eyes. So that does create variation in his offspring. So that is Mendels Law of segregation. The alleles segregate or separate into gametes, haploid gametes. And then the second thing mentioned is, if you see right here, independent assortment. So in independent assortment, this is where alleles of 2 or more genes are so here gene A and gene B are basically don't influence each other and how they end up in gametes. So here only I have 3 chromosome pairs, but the 2 genes, A and B, can line up, the chromosomes can line up on different sides of that metaphase plate in metaphase 1 of meiosis, which is the process of making gametes. And so all these like different possibilities of how our chromosome pairs align creates, in humans, we have over 8 million different combinations. So that would create lots of variation in our offspring. If you want a more detailed explanation of independent assortment, check out my meiosis video. But yeah, so independent assortment is going to create lots of variation in the gametes and therefore offspring. And then the third one is random fertilization. Now, I don't think I ever directly mentioned this in any of my videos, so here we go. So when we talk about fertilization, you have, like in humans, the female releases 1 egg, unless it's twins, you know, maybe there's 2 eggs or triplets, 3 eggs, but generally you're going to have 1 egg. But in the male contribution, there's going to be hundreds of millions of sperm. Now those sperm are made from meiosis with crossing over and independent assortment. So all of the sperm are unique, right? The genetic combinations within that sperm are all unique, and it's random which sperm is going to fertilize the egg. So therefore, after multiple offspring are made, you're going to have variation with different combinations of genetics in the offspring. So to summarize this essential knowledge, segregation, independent assortment of chromosomes, and fertilization result in genetic variation within populations. Okay, so then the second of the 3 like essential knowledges, or what I call them standards, for topic 5.6 is the chromosomal basis of inheritance. So basically, what this means, this chromosomal basis of inheritance, is the understanding that our traits and our genes are passed on from parents to offspring by chromosomes. Chromosomes are like the vehicle or the thing or the factor that actually pass traits on. And that's what that means. So this is just a screenshot from one of my earlier PowerPoints to show, like, as we make our gametes, and then the gametes fertilize, and now we have an offspring, and it's chromosomes that carry our genetic information. Then our last topic in this, or last standard or whatever in this topic 5.6 is this. It's about human, like, diseases or disorders. So human genetic disorders can be attributed to the inheritance of a single affected or mutated allele or specific chromosomal changes such as non-disjunction. So the first one we'll talk about is sickle cell anemia. So when we talk about a single affected or mutated allele, we're talking about one particular locus on a chromosome, like one particular location or gene. So in sickle cell anemia, it is actually due to a missense mutation. So like one single change in the DNA sequence. So a single affected gene led to the misfolding of hemoglobin. And when the like sickle cell version of hemoglobin folds up, it forms a straight chain, which causes the blood cells to form a sickle shape. So that is an inherited disorder that is due to a single gene mutation. Another example of this would be Tay Sachs. So Tay Sachs is actually, again, due to a single mutation in one gene, like a point mutation, that leads to lysosomes in neurons and brain cells malfunctioning. And basically, it's missing a hydrolytic enzyme that hydrolyzes and breaks down and digests fat. So a person who is homozygous recessive for Tay Sachs, their brain will actually, over their first couple years of life, will accumulate with undigested fat, and that leads to paralysis, blindness, deafness, and eventually death, as their brain is no longer able to work as it fills up. So a very devastating trait to inherit. And then we have another inherited disorder, Huntington's disease. Now, Huntington's disease is actually inherited as an autosomal dominant trait. And so with this, it isn't due to a single point mutation, but rather a duplication. There's a certain sequence in the DNA that is replicated over and over and over. So a person with 35 or more of the CAG repeat, so it's basically like you copy and pasted multiple times a section, so their gene is actually very long, and then the protein is very long. It actually causes the neurons in the person with Huntington's to degenerate over time, to break down and no longer work, and so therefore it has debilitating consequences. Now, the issue with Huntington's disease, however, is that it is adult onset. The symptoms of Huntington's disease don't show up until you're already past reproductive years. So you start to see symptoms between 30 and 50 years old, but by then you've already had offspring. So even though it's a dominant disorder, a dominant disease that you can inherit, you could be carrying that dominant allele and not know it until you're already older and past reproductive years. So therefore you can still pass it on. Thankfully, we live in a time where we can have our DNA analyzed, and now we can be checked to see if we are carrying, or not carrying, if we do have the allele for Huntington's disease that we inherited from parents or not. So yeah, thankfully we live in the modern age, and we don't have to guess. Okay, and then now we're going to move into talking about traits on X chromosomes, so sex-linked traits. So here, when we talk about, I don't really consider colorblindness to be a disorder, but it is a human condition that can be inherited, right? So females who are homozygous dominant or heterozygous would not be colorblind, but if they're homozygous recessive, they are colorblind. And then males are called hemizygous. They only have one X, and so therefore they only have one allele. So males tend to be colorblind more often than females, because they never have a chance to get that second dominant allele. But here is another example of an inherited human condition due to a single allele, on this case on the X chromosome. And then our last part of this standard is about specific chromosomal changes such as non-disjunction. So I have a whole separate video and PowerPoint on non-disjunction, but this is really due to either homologous pairs or sister chromatids failing to separate properly during meiosis, and it leads to an individual being born with an extra or missing chromosome. So Down syndrome, for example, would be an example of non-disjunction, and that's an inherited syndrome due to an extra chromosome, having 47 chromosomes. There's other ones like trisomy 18, Klinefelter's Jacob syndrome. There's a handful of other ones, but here we have Down syndrome as my example. All right. This is it for Unit 5. Great job.